Good morning, everybody. So just I was going to say reminder, but I haven't told you about this because I forgot to tell you about this seminar that I'm sure all of you will now be dying to go to because you know something about lipid A and you know something about Vibrio fischeri and the squid symbiosis. So just by random chance, this is not my guest speaker, there's a seminar this Friday at 4 o'clock in the Andrews 101 that you don't have to attend. There's no extra credit. There'll be no exam questions. Just if you're interested. It's actually Will Soto's guest and a friend of his. So I'll remind you again on Friday because you're apt to forget. So we're now going to move on in topics and look at transcription in bacteria. In part because I think you probably spent a fair amount of time talking about transcription in other biology classes and largely, possibly, spent most of your time talking about transcription in eukaryotic organisms. And transcription in eukaryotes is very different than it is in prokaryotes. As a matter of fact, we take advantage of the fact that bacterial transcription, making a messenger RNA copy of DNA, we take advantage of that being so different in bacteria that there are antibiotics that target the bacterial RNA polymerase. So bacterial RNA polymerase is sufficiently different from ours that drugs that affect that polymerase don't affect ours. And so I'll point some of those out as we go along. But in terms of one of the major differences of transcription between eukaryotes and prokaryotes is the fact that prokaryotic transcription can occur in cisterons or in polycisterons, or more likely referred to as operons. So you can have two adjacent genes. Actually, you can have 17 or 20 adjacent genes in a bacterial genome that will be transcribed into a single long messenger RNA that can then be translated separately into proteins, in this case, protein A and protein B. This is the simplest possible operon or polycistronic messenger RNA. This is a situation which really cannot occur in eukaryotic cells, and we'll talk about the mechanistics of why it is that bacteria can make operons, but eukaryotes can't. The other really important difference at the most basic level between eukaryotic transcription and prokaryotic transcription is that in eukaryotes, those two processes of transcription and translation occur in separate compartments of the cell. So transcription of eukaryotic DNA in the messenger RNA occurs in the nucleoplasm within the nucleus, and then the messenger RNA is transported to the cytoplasm for translation. In bacteria, because obviously there is no nuclear membrane, transcription and translation occur in the same compartment, and in fact, they occur simultaneously. So this piece of messenger RNA that's been copied from this bacterial genetic material here, gene A and B, is actually being translated into protein A before this entire piece of messenger RNA has even been synthesized. So transcription and translation occur simultaneously in bacteria, but they occur temporally separated and spatially separated in eukaryotes. And then the other major difference between transcription in eukaryotes and prokaryotes is the presence of introns within eukaryotic systems. Bacteria, thankfully, don't have introns, so we don't have to worry about RNA splicing and processing for the most part. There are a few archaea that do occasionally have introns, but for the most part, when we're talking about transcription in prokaryotes, bacteria or archaea, we can largely forget about these introns that have to be excised in eukaryotic RNA prior to being transported into the cytoplasm. So these are some of the basic things that separate transcription in these two domains. And so as you look at how transcription occurs between these, when you look at the actual enzyme RNA polymerase, you can see profound differences between RNA polymerase among the three domains of life. So eukaryotes have a very complex structure to their RNA polymerase assemblage. Partly because eukaryotes have so much larger a genome, they have a very elaborate arrangement of genes and gene families that make up 3 separate RNA polymerases. And they've actually used these 3 polymerases to divide the labor up within the cell. So for instance, the majority of messenger RNA that's made in your cells and mine is done by eukaryotic RNA polymerase II, and it has these subunits. This is only to show you the complexity of this protein. The ribosomal RNA that's synthesized or transcribed within eukaryotic cells is done by a completely different RNA polymerase. It may share some of the same subunits, but it has some unique subunits. But so there is an entire RNA polymerase with several unique proteins that is dedicated simply to do nothing more than to transcribe ribosomal RNA, and the same thing for transfer RNA. Now, that makes sense in terms of the complexity because eukaryotes are more complex than archaea or bacteria. But if you next look at archaea, that domain of the prokaryotes, you can see that there's some similarity in the transcription between archaea and eukaryotes. And that makes sense because remember that split between bacteria and archaea that occurred was later followed by a split from the archaea that led to the eukaryotes. So this is one of these situations where archaea are actually more closely related to eukaryotes than they are to bacteria. So even though we call both archaea and bacteria collectively prokaryotes, that's an artificial term. They're not more closely related simply because they're prokaryotes. And this is the first really good example, I think, of that. Because what we tend to think of is, well, they're small, unicellular prokaryotes, they must be more related. But in reality, you can see when you look at the architecture of the single RNA polymerase that archaea has, it has a much more complex arrangement of proteins, much more similar to the eukaryotes than this very simple, or apparently simple, bacterial RNA polymerase shown on the left. Where in the bacterial RNA polymerase, these are very different molecules. This is part of the reason why we can treat bacterial infections with antibiotics like rifampicin. We'll come back to that in a minute. Rifampicin inhibits the RNA polymerase of bacteria, but it won't do a damn thing to an archaea RNA polymerase or a eukaryote because they're so different. Yet, they have to accomplish the exact same things. So if you look at the subunits of bacteria, you see there's a little bit of a typo here. This is not beta and beta. These are 2 separate units. They're actually called beta and beta prime. And then there are 2 subunits of alpha. These are actually identical to one another. And then there's this mysterious little one, omega, which we're really not going to talk about because it's a big area of controversy. Nobody really knows what in the hell it does. We can eliminate it from a bacterial genome, and the bacteria can still transcribe DNA. The regulation is off a little bit. But omega is kind of it's the ugly stepchild of RNA polymerase. No one really wants to work with it because no one can really figure out what in the hell it does. Now, if you look cartoon-wise, so I love colors and I like cartoons, but this kind of shows you a little bit more about the complexity of eukaryotic RNA polymerase and the regulation of transcription. So part of the way that we adapt as eukaryotes to different environmental conditions is that we transcribe certain genes only at certain times and not others. So if you have this RNA polymerase here, this could be RNA polymerase 1, 2, or 3. It really doesn't matter. It has an enzymatic function. It has a motor. It has a lot of general transcription factors, so you see the complexity of it. It's binding to a DNA sequence here called a promoter, which has a Tata box. But in terms of regulation, you can see, and I'm going to contrast this with the next slide with bacteria, that these regulatory spaces or cis-acting these sequences of DNA that bind to regulatory proteins can be hundreds or thousands of base pairs upstream. The eukaryotic genome is so much larger than bacteria that the sites where regulatory proteins bind can be thousands of base pairs away, but still have these proteins interact with RNA polymerase to affect transcription. And in fact, this cartoon doesn't show it, but there can be regulatory sequences downstream of where the gene starts its transcription, which would be here, which can also affect transcription. So this is just my way of giving sympathy to those of you taking cell biology, because when you're learning about transcription in cell biology, you have a hell of a lot more complex task in front of you than you're going to have in microbiology when we talk about bacteria. Yet you have to remember that even though bacteria are much more simple, they have to accomplish the exact same things in terms of gene transcription and regulation that eukaryotes do. They just have to do it with a simpler body plan. So it's simpler, makes it easier for you to remember for exam time. So this is just a generalized scheme of the RNA polymerase generalized for bacteria. So let's ignore this red box here or this blue box below. Those are regulatory factors. Those would be transcription factors that would alter gene expression. We'll talk about those in a minute. But here is the RNA polymerase of bacteria in all its glory. So it has beta and beta prime subunits of proteins. The omega is not shown in this cartoon. And then there's the two alpha subunits. So what this is showing here is alpha carboxy terminal domain, so CTD stands for carboxy terminal domain, as opposed to the alpha subunits which have an amino terminal subunit domain, which is the alpha NTD. So these are two identical proteins that interact with the beta and beta prime. And at some level, this very extended protein, the alpha subunits, the carboxy terminal domain, can interact with transcription factors. So regulatory proteins that actually perhaps increase the amount of transcription. So this carboxy terminal domain can interact with proteins upstream, but not very far upstream. So this is one of the other things about bacteria, because they're small, because they have smaller genomes, their regulatory sites, their DNA sites that bind to regulatory proteins, tend to be less than 100 base pairs upstream from the RNA polymerase binding site. And they're very seldom, if ever, downstream. So there's some basic differences between how bacteria regulate their RNA synthesis. And so in this top one, this red sphere-ish type thing here is a more common body plan for how a transcriptional factor, like an activating factor, can bind. It will bind more proximal to where the RNA polymerase binds, so that there's actually a physical interaction between the proteins. So this would likely be an activator protein that would help this RNA polymerase become stably interactive with this promoter region. So what's also being shown in this cartoon is a subunit that was not listed in that previous table. So there was one, if you look back at this table, you'll see beta, beta prime, alpha, two alphas, and omega. But when you look here, you see this brown one down here in contact with the DNA, sigma. The authors of that previous book view sigma as a transcription factor rather than a subunit of the RNA polymerase. I tend to think of it as a transient RNA polymerase subunit, because this sigma subunit is only going to function, and so only going to be part of the RNA polymerase when RNA polymerase binds to its promoter region. So by promoter region, by definition, I'm sure you all remember this, is that a promoter is a region of DNA that binds RNA polymerase to initiate transcription. So shown here in these two purple boxes, collectively those two purple boxes would be referred to as the bacterial promoter. We'll talk about these in more detail in the next slide, but here is the start of transcription shown by this bent arrow. So that's a standard signpost for where transcription starts when you see a bent arrow like that. And that's called the +1 site. So that's the first point at which messenger RNA would be synthesized. But what this sigma subunit does is it gives this RNA polymerase the specific ability to recognize that promoter. The RNA polymerase by itself cannot recognize the proper place to bind. What the remaining portion of this RNA polymerase does is synthesize RNA. That's its job, and it does that very well. And it does not need sigma in order to synthesize messenger RNA. So that's why I refer to sigma as a transient subunit of the RNA polymerase. It's necessary only at the point where the RNA polymerase needs to locate where to start. Once transcription starts and this RNA polymerase begins to move to the right, sigma will be lost. Sigma will now be recycled onto another RNA polymerase to hopefully start transcription again. But the sigma is just here. S in sigma stands for specificity. That's why they called it sigma factor. The S for specificity. It allows for the specific binding to this sequence shown in purple and this sequence shown in purple. So sigma does that, and then its job is done. It's lost, and this RNA polymerase will continue to the right. So those two purple boxes that I was just showing you are still kind of shown in purple in this next slide. But here they're called by their more proper names, -10 and -35. You will still occasionally see the -10 box, the -10 half of that promoter, referred to by kind of a historic name occasionally called a Pribnow box. I don't know who Pribnow was, but I'm sure it's named after some famous early microbiologist. But that's what this promoter is. It's a combination of -10 and -35. These are about 6 base pairs in length each. They're hexamers. And this is what sigma binds to. So sigma binds at the level of the DNA to these -10 and -35 to begin transcription at this +1 site or transcriptional start site. So remember that I already mentioned that bacteria use a single RNA polymerase. But they have a variant subunit, sigma. Remember that fact for a moment, and then let's reflect back on the fact that eukaryotes have this much more rich tapestry of RNA polymerases. They have specialized their transcription into, or divided the labor, if you will, among 3 different very complex RNA polymerases, 1, 2, and 3, each with about 10 subunits. Bacteria accomplish the same division of labor simply by using different sigma factors. So you can have one sigma factor, one protein, which would allow the RNA polymerase to bind to one class of promoters. So a class perhaps of promoters that were involved in motility. But then when the motility genes aren't needed to be expressed as much, the bacteria simply uses a different sigma factor, substitutes in a different sigma factor, and now the RNA polymerase will bind to a different class of genes. So bacteria have done the exact same thing in terms of division of labor among the genes that need to be transcribed that eukaryotes have done. They've just done it in a completely different manner, a more conservative manner, conservative in terms of genetic material. And this makes perfect sense because they have less genetic material and less energy budget to play with. So they have to have accomplished the same things that eukaryotes have done, but in a more conservative manner. So the most common bacterial promoter, the one that gets most of the press, is referred to as this -35 and -10 hexamers. But there are other promoters, and I'll show you some in a little while, that are slightly different. So some are at -12 and -50, depending on what the sigma factor binds. But this -10 and -35 is the most common type of promoter in E. coli. And so we know most about transcription in E. coli, we compare everything to this. About 70% of all of E. coli's genes have a -10 and a -35 promoter. So they're about 70% of E. coli's genes are being transcribed under the control of one sigma factor. But that leaves 30% of the genes to be divided among different sigma factors, so they can be expressed differentially at different times and different needs of the cell. So bacterial RNA polymerase begins transcription of messenger RNA at this +1 site. So what's shown here in yellow below this is the messenger RNA. And so you'll notice that it's lining up immediately under the +1. Most messenger RNA in bacteria begin with a purine, either guanosine or adenine. I don't know why that's important, but most messenger RNAs begin with one of those 2 nucleotides. And then the first region that's present on that messenger RNA, and this is an absolute requirement, is there must be what's shown here as a leader piece of messenger RNA. I prefer the term, because this is the term that's used in eukaryotic molecular biology, of a 5' untranslated region, or 5' UTR. Because 5' UTRs are very common and they're very elaborate in eukaryotic cells. Some eukaryotic genes have immense 5' untranslated messenger RNA that fold into beautiful secondary structures. Bacterial 5' UTRs, while they are always present, there must always be a 5' untranslated region on messenger RNA. They're usually 100 nucleotides or less. But the reason that every piece of messenger RNA that's encoding a protein in bacteria must have this leader or 5' untranslated sequence is this Shine-Dalgarno sequence. Shine-Dalgarno sequence, or you'll usually just see it as SD, or more frequently you'll see it as ribosome binding site, or RBS. That's what Shine-Dalgarno sequences do. This is the sequence that will dictate a ribosome to load at this point in particular, inside the 5' untranslated region, and then begin to translate just about 8 or 9 nucleotides downstream at the start codon, which is usually AUG. But not always, and we'll talk about that. So a ribosome would bind here. Now, I'm going to come back to this point because it's always good to be able to compare and contrast eukaryotic transcription with bacterial transcription. Notice that in bacterial transcription, the ribosome is binding internal to the messenger RNA. This is a very different situation than the way ribosomes load onto eukaryotic RNA. In eukaryotic RNA, a ribosome loads onto the 5' end, onto a special cap that's put on there, and we'll talk about that. So a eukaryotic ribosome can bind only at the very end, the 5' end of messenger RNA, whereas a bacterial ribosome can bind internally anytime it finds a Shine-Dalgarno sequence. This is also a very important point because this is part of the reason, maybe the whole reason, why bacteria can have operons, polycistronic messenger RNA, and eukaryotes cannot. If this piece of messenger RNA encoded 2 different proteins, the ribosome would fall off at the first stop codon, but then if there's an internal Shine-Dalgarno sequence in front of the other gene, a new ribosome can bind and begin to translate. That can never happen in a eukaryotic cell because eukaryotic ribosomes bind only to a specialized cap on the 5' end of messenger RNA. So that's a large part, if not the entire reason why eukaryotes virtually never, if ever, have operons. Okay, so here, shown now at the nucleotide level, so you can actually begin to see what these hexameric sequences of -35 and -10 look like. Imagine that RNA polymerase has bound to this. So the sigma factor recognizes this -35 sequence. This is the consensus sequence. This 6 base pair sequence is the perfect 6 base pair sequence for that general sigma factor. Remember I said that 70% of E. coli's genes are transcribed by RNA polymerase containing 1 sigma factor. That majority sigma factor binds best to TTTACA, and so these are not perfect sequences, I'm sorry. But here's the -10 sequence. So RNA polymerase binds best to TAT-AAT, so this is not the exact perfect sequence, but RNA polymerase binds to this -10, this -35 to initiate transcription. And the way that transcription is initiated once RNA polymerase binds to this is there's a melting of the DNA. The RNA polymerase functions as a helicase. It's able to break the double-stranded bonds between this portion of the DNA, and it forms what's known as an open complex. So this local melting provides a single-stranded template onto which you can begin to synthesize messenger RNA. So the blue strand here, the template strand, is actually the template that gets copied. That's where complementary nucleotides are added. So you see the first nucleotide added on here is a guanine complementary to this cytosine. So again, this is a purine nucleotide. This is the most common nucleotide that bacteria use to initiate transcription. And you can see it still has the 3 phosphates on it. And then adenine is added, and then a uracil will be added here. So this is the beginning of the transcription. So RNA polymerase has begun by opening up the DNA and is beginning to put in complementary nucleotides, ribonucleotides at this point, because it's making messenger RNA. But at this point, sigma has filled its role. It has found this promoter. It's bound to this TTT-ACA and this TAT-GTT and allowed the RNA polymerase to begin to transcribe. But now, sigma is actually working as an anchor because it's kind of got a stickiness to it. It's designed, it's evolved to bind to these sequences. So RNA polymerase, as long as it's got sigma on it, is really kind of stuck. It's trying to move to the right, but sigma is holding it back. So what happens next is a process known as promoter escape, where sigma is kicked off. It is physically ejected from the RNA polymerase. So now the RNA polymerase has nothing holding it back. It can just slide on down this DNA, making a messenger RNA copy. It's lost sigma, which is good because now sigma can be recycled. It can bind onto another RNA polymerase that doesn't have a sigma and initiate transcription once again. So sigma is lost, and the remaining subunits, which is referred to as the core enzyme, continues to travel along the DNA, synthesizing messenger RNA. This shows it a little bit better. Yes, I'm sorry, Brett?
Yeah, what actually kicks off the sigma?
So I always portray it in class as if it's an active process by the core enzyme kicking it off. And I don't know if that's really true, but it's a nice image to have in your head. It may simply be that it's just kind of, you can view it as being a little bit sticky and stuck to these sequences, and so as the RNA polymerase tends to move, it just pulls the two of them apart. But what the actual mechanism for the loss of that sigma factor, I'm sure somebody knows. It just doesn't happen to be your professor. I'm sorry. So this shows it again, because the RNA polymerase in bacteria really exists in two different forms. It exists in the core form where you have beta, beta prime, and the two alpha subunits. And that's the portion that is completely competent for synthesizing messenger RNA. It needs nothing else. It doesn't even need the omega subunit, which isn't shown from this old textbook. But then it can add a sigma factor, and now this, what's referred to as the holoenzyme, is capable of finding a specific promoter. So remember, S for sigma standing for specificity. This holoenzyme is what binds to promoter sequences to allow the bacterium to dictate exactly where transcription begins. So once this holoenzyme has bound to the promoter and messenger RNA synthesis has begun, sigma exits the picture, and the core enzyme can continue to synthesize. Now, I mentioned this earlier, and I want to come back to it because this is a major point. Remembering that eukaryotes have this very elaborate 3 different RNA polymerases to do 3 major transcription jobs. Bacteria have evolved to do the exact same thing, but in a simplified manner, in a more conservative manner. E. coli, for instance, has 7 different sigma factors. Each one of those sigma factors has the ability to bind to a different type of promoter. It has one called sigma 70 that transcribes about 70% of E. coli's genomes. It's not called sigma 70 because it transcribes 70% of the genes. Sigma 70 simply means that it's 70 kilodaltons in size. It's kind of an unfortunate coincidence that it also is responsible for 70% of the transcription of the cells. But that leaves 6 other sigma factors in E. coli that can bind to different classes of promoters under different conditions. So as the bacterium needs to change its transcription pattern or makeup, it can use different sigma factors to respond to different environmental conditions. And E. coli is actually one of the smaller genomes of bacteria. I didn't say it's smaller. It's about the average size of many bacterial genomes. But Bacillus subtilis, a bacteria that you've all looked at in the laboratory, which has a larger genome and is a more complex bacteria, has 17 different sigma factors. So there are 17 different classes of gene promoters that bacterium has, that it has the ability to temporally change its gene expression pattern simply by substituting a sigma factor. It allows it to bind to a different promoter, but then the gene transcription as occurs by the core polymerase, it doesn't matter because sigma's gone at that point. So it's just another way to do what eukaryotes have already done. And so here's just another way to look at this at a more molecular level. So here's bacterial RNA polymerase, and you can see the beta and beta prime subunits here. The alpha subunits are here, alpha 1 and alpha 2. Sigma is shown in red, so there are different domains of sigma, and I'm not interested that you know these the fact that the sigma domain 2 binds to the -10, but here's the -10 6-base pair sequence shown here in yellow in DNA. So sigma is interacting with this -10 element, or the Pribnow box, and sigma domain 4, so this is the same protein, which is wrapped around here, is binding to the -35 element. And so a local melt is going to occur in this DNA just downstream from the -10, and this +1 site, or the transcriptional start site, is where messenger RNA is going to be initiated. So once that happens, sigma is going to be lost, and the remaining portion of this protein, the beta, beta prime, and alpha subunit are going to just move on to the right and to continuing transcription. Now, the promoters vary. So there is a consensus promoter. There is a very best promoter that sigma 70 can use. Remember, I said that sigma 70 was the most commonly used sigma factor in E. coli. This is the perfect -35 sequence for sigma 70 binding, TTG-ACA. I've never asked you to memorize DNA sequence. This is just for your information. And then the perfect sequence for the at the -10 for sigma 70 to bind to is TAT-AAT. So if you put that promoter in front of a gene, you're going to get transcription like a freight train. Sigma 70 is going to find that no matter what, and you're going to get a whopping amount of transcription from that. Now, one of the ways that bacteria have fine-tuned their gene expression level, because they don't need massive amounts of transcription of every gene, they need smaller amounts of other ones, is that they vary that sequence. So here is the sequence in front of the LAC operator, or in front of the LAC promoter for the lactose operon. And you can see that it has a slightly different sequence away from the consensus, TTT-ACA. If you were to modify that T to an A, you would get more transcription. Modify that C to an A, or I'm sorry, you would get down transcription. And here's the -10. This allows the bacterium to have evolved different levels of transcription from different genes to make things appropriate. So here are just a whole bunch of genes from E. coli that have been lined up. Here's -10 sequences. Here is the transcriptional start site of these. And you'll notice again that the transcriptional start site is usually a purine, guanine, guanine. There's occasionally a pyrimidine. Here's cytosine. But most of these are purine. And then you determine the -10 sequence simply by counting up 10 nucleotides from that, and that generally puts you in about the center of something that looks like TAT-AAT. Here's a perfect one right here. And that's, fortunately, right in front of a ribosomal RNA gene, so you need lots of transcription of that gene. But then you can see there's variation from these. So each one of these promoters would have what's referred to as a different strength. Transcription would occur at a different rate and to a different level in front of each one of these, dictated by how different the promoters are, both the -10 and the -35. And that's just for the sigma 70. So sigma 70 binds to these -35 and -10 sequences. But the bacterium is also producing, at the same time, other sigma factors. So sigma 32, and again, 32 just tells you the size of the protein. It's not important. Controls heat shock-induced genes. So if you take E. coli in the laboratory that we normally grow at 37 and bump the temperature up to 40 or 41 degrees, one of the things that it has to do is to change its gene expression pattern to adapt to that temperature, because proteins are now misfolding. They need to make chaperone proteins to help them through that temperature shift. So they'll now substitute in sigma 32 to RNA polymerase, and now they'll begin to transcribe genes that previously were not being transcribed, that have -10 sequences that look completely different. And then you can have ones like sigma 54, which controls genes for nitrogen metabolism. These this sigma has promoters that aren't even at -10 and -35. This class of genes in front of genes that might be involved in nitrogen assimilation or ammonia production have their promoters at -12 and -24. So depending on what sigma factor is predominating in the cell at any one time, the bacterium can change in real time its gene expression pattern simply by changing the availability of these sigma factors. So just to point out how responsive these bacteria are, because it's not a matter of if the bacterium finds itself in an environment now where it needs a different sigma factor, it doesn't necessarily have to go, okay, I'm in a new environment, I need a new sigma factor, let's go and transcribe it and then translate it, because that takes time. So what many bacteria have done is they have the ability to make these sigma factors in advance and hold them in reserve. So I just want to show you a couple of examples of how bacteria have taken some of these alternative sigma factors and managed to hold them in reserve. So this is from a paper from a few years ago, some people working with Clostridium, similar to the bacteria in our intestinal tracts, but very similar to the bacteria in soil. This is from a soil bacteria. So Clostridium, in this particular case, loves extracellular polysaccharides. And when it senses extracellular polysaccharides, it wants to secrete lots of amylases, enzymes to break down those polysaccharides to bring in maltose and glucose. But if those polysaccharides aren't there, it doesn't want to transcribe those genes. That would not be a good use of energy. So what they've done is they've evolved this thing called an anti-sigma factor. And this is a very common feature of bacteria. So they're all different from one another. The strategies are different, but they're generally all referred to as an anti-sigma factor because they have a domain, in this case on the cytoplasmic side, which has the ability to bind to this specific sigma factor and no other sigma factor, just this one. But when it holds it like this, that sigma factor is now not available to enter into an interaction with the RNA polymerase core enzyme, the alpha-2 beta-beta machinery. So these genes, these cellulosomal genes, are not transcribed because they're not necessary. Now, if that Clostridium finds itself in an environment where there are extracellular polysaccharides, this extracellular domain of the anti-sigma factor senses those. So this CBM domain, when it interacts with polysaccharides, transduces that information across the membrane. It changes the conformation of this anti-sigma factor such that sigma is now released. So there's no going and let's transcribe the gene and then translate the gene into a protein. It's already done, and it's just waiting for the right signal. So this anti-sigma factor will be released from multiple sites within the bacterium and can now overwhelm whatever sigma factor was dominating before, enter into this RNA polymerase, and begin to transcribe these genes for cellulosomal genes. So these are genes that encode proteins that are going to be exported that will break down cellulose, so the bacterium can then import it. So this is a very rapid means by which bacteria adapt. So here's another one from a bacterium called Methylobacter extortans, another environmental bacteria, that also uses an anti-sigma factor, but you can see that it's a completely different strategy. So this is the fascinating thing about anti-sigma factors, is they're generally completely unrelated to one another genetically. But they've all evolved to do the same thing, and that is to hold a sigma factor in abeyance until the environmental situation is such that the sigma factor is needed. So in this particular case, you have this Phi-R protein, which normally exists under normal conditions if the cell is fat, dumb, and happy and there's no stress going on. It exists in one conformation where it cannot interact with any other protein. During that same situation, when this Phi-R is in this inactive form, you have this specific sigma factor bound by a soluble anti-sigma factor. So this is just a protein that's now got or a sigma factor that's got a protein stuck on the side of it that it can't shake, but it can't do anything as long as until that protein leaves it alone. So what happens is that if that bacterium becomes stressed, there is a signal transduction pathway as always, that results in the phosphorylation of a protein. So this P is a phosphoryl group that's added by a histidine kinase, such that now the Phi R protein changes conformation so that it can now outcompete. So it binds to this anti-sigma factor better than sigma does. So it literally steals the anti-sigma factor from the sigma factor, freeing it up to heave a giant sigh of relief, and then bind into an RNA polymerase, find the appropriate promoters, and now activate genes which will hopefully see the bacterium through this stress. Honestly, I don't know what this particular stress is that these researchers were looking at. But this allows the bacterium to only transcribe those genes for that stress response when the stress is present. Otherwise, it just ignores those genes. So those are just two examples of how bacteria can use anti-sigma factors to control gene expression patterns. Okay, so I mentioned earlier that prokaryotes, so this occurs certainly in bacteria, I think in archaea as well, but certainly in bacteria, the production of operons. So having multiple genes on a single messenger RNA. We're going to come back and talk about the trip operon more than you ever wanted to talk about the trip operon when we talk about regulation of transcription in a little while. But for the moment, just know that these are 5 genes in tandem on the bacterial genome that encode all of the encode all of the proteins necessary for the bacterium to synthesize the amino acid tryptophan. And now the bacterium doesn't want to synthesize tryptophan if it's got plenty, and it needs to transcribe it in a hurry if tryptophan gets low. So being able to regulate the tryptophan operon is very important for these bacteria. But in terms of how these operons work and why they work in bacteria and not eukaryotes is, so you have RNA polymerase binding to the promoter and beginning the transcription of trip E through trip A, all into one messenger RNA. Now, in each case, you have a start codon, and at this point, at the messenger RNA, remember start codon is where a ribosome begins to synthesize proteins. And then there's a stop codon shown in, appropriately enough, in red. So the ribosome will stop and disassociate at that point. Now, this is, again, points out why eukaryotes cannot do this. A eukaryotic eukaryotic ribosome would be able to bind one time and one time only to this 5' end, and then only if it has a special cap. But it binds to this 5' end, and it would begin to translate at that start codon. It would hit this stop codon, and the ribosome disassociates. And that's fine. It made trip E. But trip D could not be made because a eukaryotic ribosome cannot bind internally in this messenger RNA to transcribe trip D. This is why it's important that you understand these Shine-Dalgarno sequences. So these Shine-Dalgarno sequences, there will be one in front of each one of these genes. That allows a ribosome to bind at each one of these sites. Ribosomes in bacteria bind to the Shine-Dalgarno sequence because remember that ribosomes have RNA inside of them, 16S and 23S RNA, as well as proteins. In bacterial ribosomes, there is actually a base pairing that occurs between the ribosomal RNA and the Shine-Dalgarno sequence. That's what initiates the binding of a bacterial ribosome to RNA. That's very different than a eukaryotic system. There is no base pairing between the messenger RNA of the eukaryotic message and the ribosome in the eukaryotic cell. It's all based on a special cap put at the 5' end. So you can have a ribosome now bind in front of trip D, in front of trip E, C, B, and A, and to translate them into all of these proteins that make up the pathway, the biosynthetic pathway for tryptophan biosynthesis. So this is the arrangement in a eukaryotic system. So here would be base 1 and base 2. This is the beginning of the messenger RNA for a eukaryote. So here are the 3 phosphates that remain on the first base of that messenger RNA. In eukaryotes, there is invariably a 7-methylguanosine cap put at the 5' end. It can go no place else. The only place that this can be put is on the 5' end. And it's this cap which allows ribosomes in a eukaryotic system to bind. and to begin to translate. Once that ribosome falls off, the only place that the ribosome can bind again is at that 5' end. So this is why it's important to understand this system, because eukaryotes cannot do operons. And we have 5 minutes left, and I could launch into transcriptional termination, but I feel like I have worn out my welcome. So I'll see you guys on Friday or tonight for the exam, or sometime throughout the week, whenever you're taking the exam.